Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on the human nervous system and in particular on the control of body temperature. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hi all, and welcome to tutorial 4 of 4 on the human nervous system, in which we're going to be looking at the control of body temperature. So in our previous session we had a look at the eye and how its different structures contribute to its function. And we also had a look at the topic of accommodation, and that was actually a really detailed tutorial and a really useful one too for the exam, so do go back and watch it if you haven't seen it yet. So today we're looking at the control of body temperature, so this is all about homeostasis, and we're going to have a look at the responses to increases and decreases in body temperature, and finish that off with looking at some cooling curves. So body temperature is monitored and controlled by the thermoregulatory centre in the brain, which is found round about here. And so the hypothalamus is what monitors body temperature. And a change in temperature is detected by the thermoregulatory centre in the hypothalamus. And this centre detects blood temperature in the brain using receptors and also gets feedback from receptors in the skin. So going into the thermoregulatory centre, we're getting receptors in the hypothalamus, which are sensing your central temperature, and we're also getting receptors in the skin, which are sensing your peripheral temperature. And they are sending impulses, nervous impulses, to the thermoregulatory centre, which is going to combine all this information. So the skin has many temperature receptors, and so this is going to allow thermoreceptors to send feedback to the hypothalamus, and this is really, really important in making sure that the body temperature stays around about 37 degrees. And just remember that all of this feedback is going to contribute to keeping a constant body temperature, which is what homeostasis in terms of temperature is all about. So 37 degrees is our optimum temperature for enzyme function. And so at 37 degrees, your enzyme is going to work at its optimum, so at its best. And homeostasis is working towards making sure that we stay at this temperature. If the temperature increases greater than this, enzymes are going to be denatured. And similarly, if the temperature is lower than this, you will also get enzyme denaturation. And we explored this in a previous tutorial in which we looked at the specific structure of an enzyme, since it's a protein, and so it's going to have a very specific sequence. And so the formation of its bonds is also going to be very specific. So as you start to increase or decrease temperature, you're going to get changes in the specific structure of your enzyme, especially its tertiary structure. And you're going to get breakage of the hydrogen bonds, which are fairly weak, and therefore this is going to change the shape of your enzyme and accordingly change the shape of its active site. And this may mean the substrate is no longer be able to fit into the active site, which means that the reaction can't be catalysed. So if you're a bit confused about this, do go back and watch the video on how enzymes work. So you may notice a pattern. So responses always follow the receptor, control centre, effector pathway, and we explored this in the first tutorial in this series on the nervous system. And the thermoregulatory circuit is no different. It involves the brain, but just keep in mind that this is still subconscious, so it's not being processed consciously. You're not aware of your own body temperature changing, and you're not aware of carrying out effector mechanisms to regulate your body temperature. So for example, if the temperature is too high, it's going to first of all be detected by a receptor of some kind, and we know that your skin has receptors, so let's say it's detected by a skin receptor. You're then going to get nervous impulses transmitting this information back to the thermoregulatory centre in the hypothalamus. So we call that feedback, because your receptors are feeding back to your regulatory centre. We're then going to get effector mechanisms occurring. So when your temperature is too high, these are going to be your 3 effector mechanisms. sweating, skin hairs lie flat, and vasodilation. So let's take a look at this in more detail. So you're too hot, which means that you're going to try and lose heat from your body. So first of all, we do that by vasodilation. So this is where your vessels, so your vaso, dilate. So the blood vessels near your skin dilate, which means they get wider. This means that more blood is going to be pumped through these vessels close to the skin, and therefore more heat can be lost through conduction to the environment, rather than your hot blood running through deeper vessels away from the surface where they're less likely to lose their heat. So look here how your blood is going to pass through these dilated vessels and therefore allow heat loss to the environment. Also, sweating occurs when you're hot, and so your sweat glands open up and cause our skin to release more sweat. And when there is more sweat, more heat is going to be lost to the environment via evaporation. And this is because liquid sweat absorbs heat, which increases the kinetic energy of molecules enough to overcome the forces of attraction between them, therefore enabling the liquid to evaporate into a water vapour and allowing heat loss through radiation and convection. And skin hairs also lie flat, so your hair erector muscles relax, therefore causing the hair on your skin to lie flat. And this means that it traps less air, less hot air, close to the body, which therefore allows more heat loss. So if you're ever asked this kind of question in an exam, just think, the person is too hot, they've got to try and get rid of heat from their body. So just think from first principles how you could do this. So then you would come up with vasodilation, sweating, and skin hairs lie flat. So although it is a case of a lot of memorisation when it comes to exams, you can often work it out from common sense. You've just got to try and think of the ways in which your body may try and lose heat. Or you can even put yourself into the situation and think about what happens to yourself when you get hot. And actually vasodilation can be seen on yourself because when you're hot you might go quite red, and that's because your vessels under your skin surface have dilated. And therefore you're going to get more blood, more volume of blood passing just underneath your skin, which is why you get that kind of red flush when you're hot. And so now let's have a look at a practice question. So explain fully why we have to drink more water on a hot day. So this is the first answer that was given. We drink more on a hot day because we sweat a lot more than we would on a cold day. Now this is perfectly right, but where's the detail? It's worth 3 marks, which means you've got to try and get at least 3 valid points in there, whereas I can only really see 1 valid point in here about sweating more on a hot day. So the examiner would say this answer is too brief and doesn't explain the mechanism of sweating, which I've just told you, so you should try and put that in. Instead, let's have a look at this more improved answer. So explain fully why we have to drink more water on a hot day. So your first mark would be, on a hot day, our skin and hypothalamus receptors detect a rise in body temperature. Number 2, this rise in body temperature is fed back via nerves to the thermoregulatory centre in the hypothalamus. And number 3 is then your effector, so the sweat glands. So let's just take a look. So I really like the way that the person has done this answer because they've talked about the sensing. So this is the sensory part of your pathway. Then this is the feedback to the CNS. And this is your effector mechanism. And when you've got a question asking about something like thermoregulation or homeostasis, and it's worth 3 marks, we know that we can break down our sensing affecting mechanism into sensory, transmission to the CNS, and then your effector mechanism. And they've done this really well because they've said that we have receptors in both the skin and the hypothalamus. And if you go back to that diagram that was on one of the first few slides about how skin and hypothalamus receptors feed into the thermoregulatory centre, they've said exactly that in their answer. And it's these that are detecting the rise in body temperature, and they're feeding back to the thermoregulatory centre in the hypothalamus. And the hypothalamus in turn is going to stimulate sweat glands to secrete sweat onto the skin, which increases heat loss. And therefore, because the body is losing water via sweat, we're going to need to take in more water. So this is a really good answer.
So when you see a question is worth 3 marks, do try and get at least 3 points in. Even though the original answer we saw was correct, it just wasn't detailed enough. So, now on to our last point. So let's have a look at responses to decreases in temperature. So again, they're going to be detected by skin receptors. We're going to get feedback to the thermoregulatory centre in the hypothalamus, and then we're going to get our effector mechanisms. And just notice how we've got our stimulus to receptor, to CNS, to effector. And this is the way that the student in the previous question structured their answer. So do try and keep this structure in your answer. So this is the way it flows. So perhaps you could draw this out in your revision notes too. So when it's too cold, you're going to get the opposite to when it's too hot. So you're going to get vasoconstriction. So less blood is going to be able to pass into the skin because the vessels are smaller. So we're going to get less heat loss to the environment. Also shivering, we're going to get, instead of sweating, we're going to get shivering. So the skeletal muscles contract and relax very quickly. And we know that when a muscle contracts, it's going to release energy in the form of ATP, but also there's going to be some energy being released as heat energy. And this is going to warm the person. And we're getting this, basically, the more the muscle contracts, the more respiration takes place, so the more energy lost as heat. And therefore, you shiver when you're cold because it essentially generates internal heat. In the form of energy, of course. In the form of heat energy. And then the opposite to being too hot, so you're going to get your skin hair standing up, which causes the hairs to trap more air near the skin, therefore leading to less heat loss as it provides insulation. So shivering happens in very cold situations, while vasodilation and hair standing on end tends to happen when the temperature drops a little. So shivering is a marker of more extreme cold, perhaps. So now let's take a look at a cooling curve. So you might be given a cooling curve like this and asked to describe the changes happening in the body at different stages. So initially you would get sweating because, oopsie, I didn't mean to draw on that. Initially you would get sweating because the person is too warm, but where it's colder you're going to get something more extreme like shivering in order to generate heat for the body. So they might give you a cooling curve like this and ask you to describe the different changes happening in the body at different stages. So as I just said, for example, during minute 0, the participants may be sweating a lot, whereas during minute 5, they may be shivering instead. Also, experiments on temperature can occur, so temperature can be measured at different places of the body, most commonly the ear, the mouth, the nose, the armpit and the forehead. And forehead thermometers and skin temperature sensors are often used in a clinical situation. So that's all for today. We've run through the area of the brain which processes body temperature and how we might carry out effector mechanisms when our body is too hot or too cold. So well done for today, and I will see you for our next tutorial.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.